Negative bacteria, the shape, the morphology, may be classified into 3 groups: cocci, bacilli, rods, and spirochetes. Spirochetes are also rods, but they have a special structure. This is the reason why we study them separately from the others. 3 important for humans: Treponema, Leptospira, and Borrelia. But we are going to study only one, Treponema. So we are here. Spirochetes. Why are they so special? Because of the structure. They have flagella, but their flagella are very special. And why are these flagella so special? Look, on the left side, we have the flagella that you already know. The flagella that you already know are like this. This is Pseudomonas aeruginosa, for example. This is Salmonella. These are the flagella that you know. We call them exoflagella. Exo means outside, because these flagella that you already know are found outside. However, spirochetes also have flagella, but these flagella are different because these flagella are found inside the cell. We call them endoflagella. Endo means inside. So look, endoflagella are found inside the bacteria here. They are found between the cell membrane and the outer membrane. Spirochetes are gram-negative rods, so they have 2 membranes, the cell membrane and the outer membrane. Endoflagella are found between both membranes. This is the same. I'm going to make it bigger. This is the cell membrane. This black line is the peptidoglycan. And this is the outer membrane. Flagella are found here. And these flagella are found inside the cell. These flagella also move. These flagella rotate. When these flagella rotate, the bacteria can move like a corkscrew. This is a corkscrew. The movement of this bacteria is the same movement as this spool. So these bacteria can move through viscous solutions because of the endoflagella. This is a picture of spirochetes using electronic microscopy. You can see the shape, the spiral shape. It's not possible to observe these bacteria using optic microscopy. It's not possible. They are gram-negative, yes, but we can't see them because they are very thin, very, very thin. Because they are so thin, it's not possible to observe them using microscopy. This is a picture using electronic microscopy of Treponema. Spirochetes may be found living in the environment. Some of them are normal flora, and very few are human pathogens. These are the 3 human pathogens: Treponema, Leptospira, and Borrelia. We are going to study 3 of them. Sorry, we are going to study 2 of them. We study Treponema pallidum and we study Leptospira interrogans. We start with the first one, Treponema pallidum. Treponema pallidum is the agent of syphilis. The name of the disease is syphilis. Syphilis is another sexually transmitted disease. It was discovered 500 years ago. Treponema pallidum cannot grow in culture. So we know very few things about them because we cannot grow them in the laboratory. It's very difficult to understand syphilis. You will study in detail in internal medicine, but here we are going to explain some things. So look, Treponema pallidum cannot grow in culture. So we can't study Treponema pallidum in the laboratory. It seems that most damage depends on the host immune system. And the pathogenesis is difficult to understand, but we are going to try. So I am going to explain the pathogenesis of syphilis from exposure to disease. Before we start, syphilis is a sexually transmitted disease. The most common way of transmission is sexual transmission. So we start with exposure. Exposure through sexual contact. What happens? After 10 to 19 days, primary syphilis appears. Primary syphilis is called chancre. The chancre is a local disease, not painful. So the chancre is a painless ulcer. This ulcer in the genitals heals spontaneously in 4 to 6 weeks. So what's a chancre? This is a chancre. I show you the pictures. This. For example, this is a chancre in the penis of a man. This is primary syphilis. Primary syphilis is painless. This is another chancre. Painless. Or here, this is a woman. This is a chancre. I go back to the pathogenesis. So that is the chancre. And the chancre will heal spontaneously. Remember, the chancre is a local disease. However, after 4 to 10 weeks, syphilis becomes systemic, and patients may suffer secondary syphilis. Secondary syphilis is systemic. Patients with secondary syphilis may show a rash in the whole body. This rash may also heal spontaneously. What is secondary syphilis? This is an example of secondary syphilis, rash in the whole body. Also in the palms and the soles, both, in the whole body, like this one. Even in the tongue, this is the tongue. Secondary syphilis is a systemic disease that may be found in the whole body, also in the palms and the soles. Secondary syphilis. Secondary syphilis may also heal spontaneously. After secondary syphilis, patients may suffer latent syphilis. So this is latency. The bacteria becomes inactive. We have 2 stages of latency: the early latency before 1 year, or the late latency after 1 year. What happens after that? During latency, the infection may be cleared by the immune system. However, in some patients, the disease may appear later as tertiary syphilis. Tertiary syphilis is very severe. Even more, some patients may suffer secondary syphilis again, about 24% of patients. The most severe patients are patients who suffer tertiary syphilis. Tertiary syphilis is the most severe. Tertiary syphilis may infect the central nervous system, or the cardiovascular system, or gummatous syphilis. These are the most severe manifestations of syphilis that we can't study in detail because we don't have time. You will study tertiary syphilis in internal medicine. For example, this is an example of gummatous syphilis. This patient, this one. This is a patient with cutaneous